Good morning, everybody. Welcome to another Q&A session in this wonderful second day of the conference. We had three great presentations by Kate, Beth, and Juliette. So I will be welcoming questions now here on Zoom. Oh, sorry, you don't have the Zoom there. So here on the Q&A live session, and I will also be collecting some questions from the presentation chat page. So does anybody has any question? Okay, so I think I will start with one question from Eddie from North Carolina State University from the presentation chat. So thank you, Juliette. Did Georgie was take into account kinship among individuals? How do you take into account multiple testing when you select PEAs? Many of the effects are not different from zero.
So for the kinship, in fact, how the sampling was done is that we have only unrelated individuals, because in the population we sampled individuals separated at least 100 meters apart. And but maritime pine has a very strong population structure, so in the GWAS we took into account the population structure, even if in the GWAS it's very difficult to take it into account. And for the for the threshold, it's a little arbitrary. It comes from a previous studies in which in fact this is a GWAS come from a previous study, and it was an arbitrary threshold corresponding to the polygenicity that was estimated in this study. So I took the same threshold, and it was this analysis is done with the PyMass software. So it's not SNP by SNP analysis models. It's modeling groups of SNP. So like that, it took into account the multiple testing.
Thank you, Juliette. So there's now one question for Beth from Mitra Menon. Beth, wonderful talk. I was wondering if you looked at the relationship between cold injury and MCMT of source population.
So yes, that must not have been made as clear as I could have made it in the presentation. What I showed for mean coldest month temperature was for the source population or provenance. So that was what was shown. And that relationship, like I mentioned, is fairly weak, but there's definitely caveats to that, including the sampling, which is why doing some more sampling this fall would be good, because the timing of measuring cold injury really matters in terms of the cold acclimation process. So I measured it in late October, and actually between the 2 weeks that I measured, there was a sub-zero temperatures over the weekend. And so we saw a fast cold acclimation for the second week. So yeah.
Cool, thank you, Beth. Now a question to Kate. As for maritime pine, as shown by Juliette, there's quite a strong phylogeographic structure in lodgepole pine within BC. Is it planned to develop transfer functions specific to the 3 or 4 main lineages to see how much transfer functions could differ?
Yeah, that's a good question. So currently that wasn't in the plans, but I do think that that would be something that would be really useful to have in terms of the context of BC. Our only limitation, I think, in regards to that would be what we have available. The provenance test that we're looking at was initiated in the 1970s. So it would just be, you know, if we have that data available to create those functions, and I think that would be something very useful.
Great, thank you, Kate. Now a question from Tong Lee Wang for Juliette. What is the proportion of variation explained by genomic data relative to climate data in prediction of tree height?
I don't remember exactly the absolute value, but it was the variance explained by the genomic data was higher than for the climatic variables.
Okay, so a question from Victoria Sark to Beth. Beth, I enjoyed your talk. I wonder whether you would be more likely to detect provenance effects if you had also incorporated family into your model. Could you have controlled for the fact that some families within provenance have bad genotype?
Yeah, that's a really good point, Victoria. I don't have the kind of family structure within the provenances to control for that, but in the breeding families, and when I test them this fall, because I only tested natural populations for fall 2020, I will be able to control for family effects. So, noted.
Okay, so now I have a question for Kate. Kate, how did you select the climatic variables you use in your models? Why those two?
Oh, we selected the climate variables used in our models. We started by testing for correlation between the mortality and the climate variables. And then we fit the models using the climate variables that were associated with mortality the most, excluding any with high autocorrelation, or multicollinearity, I should say. And that's how we ended up with those. We also, because we're looking at including this mortality transfer function in a growth and yield model, we already have a height transfer function that uses mean coldest month temperature and mean summer precipitation. So it just ended up working out really well that those variables were most related to mortality and also lined up with the height function that we were already using.
Okay, thank you. So now I have a question for Juliette from Ian McLaughlin. Great to see you using the PEA approach and generating interesting results. Can you explain why the predictions for the Asturias side are so strong for all models and why there's little variation among models?
My interpretation is that in this site, all the trees grow very well because the conditions are very good. And so the genetic differences are expressing and are well predicted by the models. Contrary to some sites with harsh environments, and so there are strong differences between trees in the height, but there is a lot of stochasticity in the responses, and it's hard to predict it compared to in this type of Asturias for me, because of the good condition and the trees. We let the genetic differences express.
Thank you, Juliette. So now I have a question from Haley Thomas. For Beth, what do your cold hardness results mean for adaptation to climate and potential for assisted gene flow in large?
That's a great question. The data is pretty preliminary to make really strong conclusions so far, but given the previous common garden results as well, done by Gerald Rafael in the 1990s, many of the phenotypic traits that were measured had not very strong clines and not very high population differentiation, somewhere between 3% to 16%. And so there's lots of variation within populations. So I think that it suggests that there's high potential for, let's see, you asked about assisted gene flow. That I guess it would be interesting to see, and one of the reasons I'm looking into using genomic prediction is to see how, when we look at the genome compared to the phenotypic lines, if we're going to see that there's some potential or need for assisted gene flow in larch. So yeah, I'll set aside the assisted migration stuff since you didn't specifically ask about that.
Thank you, Beth. Now I have an open question to all the speakers from Brian Boltonis. So this is a general question about estimating transfer distance. What is the recommendation on how to estimate in advanced generation breeding populations? For example, is it common to include climatic information for progeny task locations where families were tested? So maybe Kate could try to answer that one, or, well, it's open for everybody, so.
Yeah, I mean, I can take a stab at it. I think it can be, or it's sort of incorporated in the use of transfer distance, climate transfer distance. So the difference between the origin and the location of planting. Yeah, I think that in a lot of cases it should be included. I think it's pretty obvious that the response is population specific, and that can often be that population can often be described by the climate that it originated from.
Does Beth and Juliette, do you have any comments on that?
Yeah, I would say that for me the genomic data are very complementary to the climate transfer distance, because for me we can't do common gardens for all species, and so genomic data could present an interest when we can't fit this climate transfer distance for species where we don't have common gardens. And yeah, and also to include the intraspecific variability at a finer scale at the level of the individuals.
Okay, thank you, Juliette. We have 2 more minutes. I think maybe Beth, you can comment on Antonio Castilia's question. So great presentation. I found very interesting that increase in the dispersion of your estimates of average cold injury with AMCMD, sorry. Do you have any idea for this increase in the variability?
Yeah, so I agree. I find it interesting that the increase in variability occurs at warmer temperatures. That could be a bit of an artifact of the sampling, given that in the first, that sub-zero temperature between week 1 and week 2. So there's more variability in the first sampled week at -15 that I analyze compared to week 2. If it's a real pattern, it might suggest that there's less selection for fall cold hardiness at the warmer temperatures.
Okay, thank you, Beth. So I think we have 10 more seconds. So I would like to thank Kate, Juliette, and Beth for wonderful answers and presentations. Thank you for all the attendees. And I see you in the next session. So if you want to keep answering the question, you can type the answers there, Beth, Juliette, and Kate, if you have more questions here on the chat. Thank you, everybody.